Okay, so last time we looked at a sperm cell and an egg cell and all that, and how they will fertilize and become this zygote that will eventually that might potentially be you. So today we're gonna learn why do they do that and what actually happens in that entire process and who made it. So we're gonna talk about this guy named Gregor Mendel, and you gotta memorize. Okay, no, you don't. But this guy is really important. You just gotta know that. You don't have to memorize his entire biography, but there might be some questions given to you in your school about him because he's really important. He's fundamental. Who is this dude? So this dude on the left, he looks pretty handsome. I know. He was born in July 20th, 1822, in Austria, which is now the Czech Republic. He was a monk, actually, and obviously he was also a scientist, or else he wouldn't be able to discover well traits and genetics. So he is known as the father of genetics. He came up with some laws and how inheritance of DNA actually work. So what did he do and how did he do that? Well, as you see, Mendel was a monk. He wasn't too rich, he wasn't too poor, but in the Augustine, well, in the management, he was the one that was managing the garden. So he was planting food essentially for those monks. So he planted a lot of pea plants. However, he noticed that he could potentially modify some traits of these pea plants. So he looked at everything on the left. He looked at how does the shape of the seed, the color of the seed, the pot form, the pot color, the flower position, and the stemlet. And there's more, obviously, but he was really an observant guy. So he came up with the idea of, hey, what if I cross-pollinated all of these, and what will happen? So this guy, he took maybe a green pea plant, and he cross-pollinated with a yellow pea plant. And he was like, what would happen? Or what if I cross a wrinkled green pea plant with a round yellow pea plant? What would happen? Obviously, he did it. So he came up with certain laws. So the first idea he came up was the law of dominance. So, as we all know, laws, you don't see those pretty much in sciences. I know you see them a lot in physics because of Newton, but you don't see them a lot in sciences, or to say in biology or chemistry in general. Because biology and chem, it hasn't been developing the fastest, and it hasn't been the most developed because physics exists everywhere. Sure, you know, you have Earth pulling everybody down. You have all of these electromagnetic force. You have those strong and weak nuclear force. You have all of these forces that's around you. It's easy to notice them. It's obviously easy to come up or to think of an idea or a theory of why things get pulled down to Earth. However, for biology, it's not the case. You're looking at genetics. You're looking at the idea of how people reproduce and what happens when people reproduce. That is a very new and fancy idea. And the thing is, we can't look at biology or chemistry unless we're getting onto an entire molecular level. And the fact is that we have microscopes, sure, but not everybody in their home has a microscope. So we don't really understand. We learn a lot about biology. We learn a lot about chemistry. But do we actually know what the hell is going on here? No, we don't. Because a very little percentage of us actually observed some sort of molecular substance. So biology, it has really minimal amount of laws. And Mendel was able to come up with 3 laws, and that was very impressive. So the law of dominance states that after you get one gene from dad, one gene from mom, one allele could potentially mask over the other. So he came up, because it got masked completely, he came up with the idea that some traits was probably dominant over the other, and some were recessive, and that got hidden. So right here we have a pure cherry, a pure red cherry. This is alright, let's say the capital A is red, and the lowercase a is yellow. So this completely red cherries, it had one A from its dad, one A from its mom. And this yellow cherry here, whatever that is, I mean, there's no yellow, is there yellow cherries? I don't know. But it got one A from his dad, one A from his mom. And when they crossed this together, so they pollinated, let's say, they cross-pollinated these together. They made they made these have some sort of hybrid, and they're able to come up with this. it got because let's say this was the dad and this was the mom. The dad passed on an A, a capital A, and the mom, it passed down a lowercase a. So when they form together, you will have one capital A and one little a. And in this case, we observe that there's no yellow here though. It's only red. So what does that mean? Red is dominant to yellow. And in Mendel's case, he used peace. So he realized, oh, wait a minute. Why doesn't yellow piece show anymore? When I mix green and yellow together, why is the kids all green? Why is the first generation of kids all green? What happened? He doesn't understand. And then later he realized, oh, maybe some things were dominant over the other, so it could potentially mask the other completely, which we have here. But now we gotta talk about the idea, what is one gene from mom, one gene from dad? So we never learned about it. And obviously the meiosis lesson it didn't tell us. So he actually came up another law that explained what this was, and it was the law of segregation. So he stated that each organism will carry 2 alleles for every single trait, and during gamete formation, alleles will separate. So you have a capital A and another capital A, let's say. Those capital A will separate. So each gamete will only carry 1 allele for that trait. So for a fully functioning human, you will always get 1 from dad, 1 from mom. Because remember, the gametic cells, it was 1 sperm and 1 egg. Though the sperm, it carried 1 allele. Because remember, alleles will separate. Each gamete will carry 1 allele. So a sperm cell carries 1 allele, and an egg cell carries 1 allele. And we're simplifying everything down because there's billion of alleles. You can't be functional. You gotta. There's a hair color. There's an eye color. There's how tall, how short, whatever. There's a lot of stuff. We're just simplifying it down. So essentially, you're always gonna get 1 from dad, 1 from mom. And here on the right, it was actually explained. So let's say we have a completely yellow pea plant, and we have a green pea plant. The yellow pea plant is dominant over green. And obviously, the dad is full homozygous Y. Homozygous just means you're the same thing. Homozygous just means you are the same capital or same lowercase. Homo is same. And hetero right here. Hetero is different. Homo, hetero. It's like homosexual and hetero. Okay, no, I'm sorry, guys. I'm sorry. So yellow passed down as Y, capital Y here, because that's the only thing it could pass on. It could. So let's say this is completely random chance. By the way, guys. So it has 2 alleles. It could either pass on the first capital Y or the second capital Y. But essentially, they're the same thing. They're both capital Ys. And same for the green. However, when they mix, now their children is going to have 1 capital Y and 1 lowercase y. So the funny thing comes in here. When this means first offspring generation. When this offspring, it tries to reproduce again, what is it going to pass on? Is it going to pass on a capital Y? Or is it going to pass on a lowercase y? So that's a 50-50% chance. Remember, guys, that's a 50-50. It has a 50% chance to pass the big Y. It has a 50% chance to pass the lower Y. So we do a test cross, or say we do a monohybrid cross. So this is the introduction to your next class. It's monohybrid crosses. You're going to learn what's happening. So let's say it's self-pollinated. So it's basically this and itself. So the genotypes is big Y, small y, big Y, small y. So you have it kind of here, it listed the parents' genotypes here. A big Y, a small y. A big Y, a small y. So this is parent number 1. This is parent number 2. And we just collect it. It's very simple. So you have big Y here. So that's a double Y. You have a big Y, little y here. That's a big Y, little y. But however, remember, yellow is dominant to green. Because the capital should always be dominant to the lowercase. All right. Whenever you guys do this question, you have to do a let statement. So I want you to always put the let statement. Let capital something be the dominant one. So you have this. So yellow, because it masks green completely. The green doesn't show. This is only yellow. So we will have 3 yellow and 1 green. However, these two, they're heterozygotes. Remember, they have 1 big Y, 1 little y. So that gives us a ratio, a 3-to-1 phenotype ratio. It means that you have 75% chance of getting a yellow and 25% chance of getting green. And now, we also have a genotype though. So we have the possibility of a homozygous yellow, or homozygous dominant. In other words, we have a 25% chance of getting a homozygous dominant. We have a 25% chance of getting a homozygous recessive. And we have a 50% chance of getting a heterozygous, or also known as carriers, because they carry a green gene. However, they're not green. But you thought this was the limit of Mendel? Nah. Mendel came up with another law that's even smarter. So he came up with the idea, hey, what if, just what if I used the raincoat and ugly looking green seed, and I pollinated with a yellow round seed? What happens? So he came up with the idea of law of independent assortment. So this isn't important as the law of dominance or the other one, or the law of segregation. However, it tells us, hey, the alleles for different traits on the gamete are fully and absolutely independent from each other. So the allele for seed color does not affect shape or whatever. So how would we be able to do a cross like this? Well, we look at the parent. We have, this is full homozygous yellow and raincoat, let's say. And this is full homozygous recessive green. Sorry, this was full homozygous yellow and round. This is full homozygous green and wrinkled. So this is a little unclear, but we will do a little bit more deeper into this. This is a dihybrid cross. Remember, you see this dihybrid cross? This was a monohybrid cross. So we're gonna. This is a full other class. We're going to do a lot of this in the future. However, today I just want to get you to be familiar with these laws. So essentially, when we use these laws, we will be able to start drawing some stuff. And these will be on your test. These will make up a good majority of them. And remember, guys, there were 5 marks, so they're really weighted heavily. And I will be starting with monohybrid crosses next class, and hopefully we'll be able to finish dihybrid in the same class, but that is on me.